Golgi bodies. This was discovered by Camillo Golgi by electron microscope. The processing or modification unit of cell is Golgi body. It has four components: cisternae, Golgian vesicle, vesicle, and tubules. The in plants, cisternae are not interconnected. That is, they are separated. They are called dictyosomes. In fungi, there will be unicisternal dictyosomes. The Golgi bodies are polar in nature in two ways. They are either structurally polar or functionally polar. Structural polarity means each cisterna has a convex side called cis phase or forming phase. Other surface is called concave, called trans or maturing phase. The functional polarity is on the basis of function. The transitionary vesicles carrying the substrate from the endoplasmic reticulum will fuse with the cis phase to empty their contents to the lumen of the cisterna. Okay, lumen of the cisterna has several enzymes to modify these molecules. The modified or processed molecules are released from trans phase in form of Golgian vacuoles and Golgian vesicles. This means that there is unidirectional flow of biomolecules from cis to trans, again cis to trans, and this continues. Tubules are penetrations or extensions arising from cisternae. Golgi bodies are surrounded by a zone of exclusion, that is, it has a part of cytoplasm that is free of ribosomes or cell inclusion. This is a example a diagram of diagrammatic representation of Golgi body. This is the cis phase and this is the trans phase or maturing phase or concave side. This is the convex side. These are the transitionary vacuoles. This is the Golgi vesicle and vacuoles. And this is the zone of exclusions without ribosomes or anything. Functions of Golgi body: synthesis of conjugate molecules like glycoprotein. It only synthesizes the three conjugate molecules. Number one, glycoprotein, lipoprotein, and glycolipid. All these three, the formation of glycoprotein is called glycosylation. Of lipoprotein is called liposylation, and that of the glycolipid is called glycosidation. Now, vitellogocytogenesis is the synthesis of Yorkin egg. In human sperm, it forms the acrosome. It is also the precure of lysosome and peroxisome. In plants, the Golgi vesicles with calcium and magnesium peptides form the cell plate or middle lamella. Middle lamella we just had study responsible for membrane flow means synthesis and repair of membrane. Membrane flow means synthesis and repair of membrane, not actually membrane flow. Okay, so now for an example how it works, RAR secretes the membrane protein, SER gives sugar lipids. Now SER gives sugar lipids which are then transformed into transitionary vehicles which are given to Golgi bodies as the cis phase, and then it releases from trans phase and reaches to the cell membrane. This is the transitionary vehicles which enter the cis phase, that is this side, and on the inside there is enzymes, and now at the trans phase or mature phase or concave phase, and procedure process will arise, and this will circular will be a circular, and this will be called Golgian vesicle, okay, or Golgian vacuole, and this would separate from the Golgi bodies and help in the formation of cell membrane. Okay, now third, suicidal bags or lysosomes. It is the hydrolytic enzyme capable of the digestion of cell or cause lysis, that is, parts of the cell, divides the cell and destroys the cell. Christian de, Christian de Ure, here the second is the capital D is silent. Christian de Ure discovered the lysosome. He said he discovered because of a biochemical analysis of the cell. Okay, just he analyzed and said there would be something like lysosome or maybe something, but actually it was discovered or seen under microscope or observed by non-cough. Not very important, but still rare and can be asked. It is smallest membrane organelle, approximate 0.5 μm, okay, in diameter. This was the size of the um, PPLO. Okay. Now, lysosome is a spherical bag having a membrane enclosing granular core. This is our lysosome. It has a membrane. On this is the cytoplasm all around. Okay, and inside it has a granular core which is acidic in nature. Granular core is hydrolytic enzyme, that is acid hydrolase. The hydrolytic enzymes are the enzymes working at acidic pH. The acidic pH inside the lysosome is maintained by continuous proton pump from the cytoplasm. Okay, it needs to be acidic here, so it is made acidic here by the proton pump from the cytoplasm. The membrane is stabilized or protected by the action of acid hydrolysis with the help of stabilizers such as cholesterol and corytosins.